So welcome everyone to this webinar. So here we have Dr. Melina Zempila. She is an atmospheric physicist and Earth observation scientist in the radiometry group at STFC RAL Space. She has more than a decade of expertise in UV and global solar radiation, radiative transfer modeling, and calibration of solar instruments. Melina has worked with STFC Food Network for several years on scoping projects, which she will talk to you about today. And she will be also lending her expertise to support our first ever STFC Food Network intern. We have asked Melina to join us today to explain how her skills has been used in agri-food projects and also to provide food for thought for future work. Before I hand over to Melina, I would just like to say that we are very keen to support projects that build on the skills of the researchers at RAL Space, like Melina. So do get in touch if you have an idea for a project, or you would like us, like to identify potential collaborators. We strongly encourage those present today to consider putting together an application for our funding call and to take part in our Sandpit event in June this year. Basically, our early career secondment researcher calls are open throughout the year, and we have a few spaces left there. So please do get in touch with us. Gareth would be able to share you the contact details, and there are details on the website as well. So yes, I would now hand over to Melina for this webinar. Over to you, Melina.
Okay. Thank you, Sona, for the introduction. Good afternoon, everyone. First of all, I'd like to thank you all for being with me today. And of course, I'd also like to thank our hosts, the Food Network Plus team, for giving me the opportunity to share part of our research. When I joined RAL Space 4 years ago, I had no previous experience in hyperspectral imaging. So you see, as Sonal mentioned, my background is in atmospheric physics. So up to that point, I had been working with ground-based solar sensors, radiative transfer modeling, and solar energy applications. So when I came in, Hugh Mortimer and Michelle Hamilton had already built the hyperspectral system that I'm going to present today and had just started working with it, that it was perfect timing for me to start building from scratch the imaging protocols and the processing chain. It definitely has been quite an exciting journey, and hopefully I will be able to share with you today all the things that I've learned so far. So I'd be happy to hear your thoughts, ideas, and concerns you may have, and of course your questions anytime during the presentation. I'm sure that we will have a constructive conversation afterwards. So I thought that it would be good to prepare this slide as a short introduction to the topics that I'm going to analyze today, just to give you a heads up on what to expect during this talk. I will start with a short discussion on the imaging technologies that are currently out there and allow us to see the world around us with a different perspective. That's in fact the concept behind the title of my presentation, because hyperspectral images give us a lot of information so we can reimagine the world as if we could see more colors in it with our bare eyes. But how user-friendly are these technologies? What are the benefits, and can they outmatch the challenges that one has to face in order to get some meaningful data? And how is it to work with such imagers? We have been developing our system for a while now, and I think that we have learned a lot during the past few years. So I'm going to try to give you some insights based on my experience. After that, I will conclude by showing some fancy, colorful images we got through our research, which I guess should be the most exciting part of this presentation. So let's begin with the discrimination between the multispectral and hyperspectral terms. Within this picture, you can see very, very quickly the main differences between the two of them. So a typical RGB imager has only 3 bands and recreates what we actually can see with our bare eyes. But a hyperspectral sensor may have like hundreds of bands, while a multispectral one usually has less than 10 bands. And this is one of the discrimination between the two of them. Another difference is that the bands in multispectral sensors tend to be spectrally wider, thus probably overlapping one another, as you can see in the figure. Oh, where's my mouse? There it is, in the figure in the middle. So this is what we actually get when we use a hyperspectral imager, and from now on, we will refer to it as a hypercube, where the 2 dimensions correspond to the dimensions of a simple image, while the third one corresponds to the wavelength or the band of the sensor. But is hyperspectral imaging as simple as it may sound, or may not? The answer I would give would be yes and no. No, just because there are so many distinctive techniques out there that one can be lost at the beginning. But when you have a well-defined project and you know what you need to do, then you just have to pick the technology that best suits your needs. And the question that comes, at least to my mind, is what should be in my checklist? What characteristics should I be looking at? When it comes to the imager, one should check the spectral range of the sensor. How many bands does it have? How narrow are they? How fast can it capture an image, which is really crucial for aerial imagery and in general when you have to deal with moving systems like a conveyor belt. And so do I need to add some optic components and how this is going to affect my images? My suggestion is to have a clear picture of what you need to do your research, contact fellow researchers who have already developed a similar system. Please contact us. We are always happy to assist, and then speak with the manufacturers. They know their products and they will probably provide some good advice regarding the choices you may have. Once you get your hyperspectral system, you gain on spectral and spatial resolution. As any other sensor, though, it requires its radiometric and geometric calibration as well in order to acquire accurate or as accurate as they can be and time-independent results. But one could be a bit flexible here and choose what type of radiometric calibration he would like to have. For example, if you convert your absolute radiance, then you won't have to perform the calibration imaging processing on every batch of data, which is something that at least we are doing when translating the raw data into reflectances. Again, there are a series of corrections and tests that should be periodically performed, and I won't go into many details here as I have some more information later on, but I need to highlight the importance of all calibration related activities, and sometimes they can be challenging for the end user. And things can get a bit more complicated when you mount your imagers onto a UAS. You can, of course, fly and get data no matter what the cloud coverage may be on the day, as long as it doesn't rain or it's not too windy to fly. It would be good to have some ground control points for georeferencing, especially if you're flying a push broom camera. And what is lacking from most of the hyperspectral systems that I've encountered so far is the correction for changes in light condition during your flight. And I will give you some more details on that just before the application section. And after wandering in this maze of calibration procedures, you finally get your hypercubes nicely organized and calibrated with the best possible accuracy you may have, and you start playing with some data analysis tools, which isn't as chaotic as it may sound, but surely takes time, especially if you have never worked with imagery before. But once again, when you use aerial hyperspectral imaging, things can get more complicated since another parameter is added to your processing chain and has to do with what we call stitching of the images. The good news, though, is that there are a lot of resources on the net that you can check out and decide which best serves you, since all commercial products provide a trial period. Here, I just added some tools that I have used for my analysis and some additional ones that I think they could be a good choice for a first-time user. So after this relatively short introduction, I think that it's now time to introduce you to our own hyperspectral system at RAL Space. It comprises of two Zempila snapshot mosaic imagers, one sensitive in the visible spectral range and the other in the near-infrared. By combining these two cameras, we get images in 41 wavelengths, while the system is designed in a way suitable for both in-lab and aerial deployment. What we actually get is a repeated pattern of spectral information over the whole image, as shown in these two figures. On this slide, you can also see the different spatial resolutions we can achieve when flying at different heights, which I think is quite impressive, as we are talking about tenths of centimeter resolution at the worst case scenario when we fly at 120 meters above surface, which is the highest altitude we can fly over the UK. And now let's dig into the calibration procedures. Here you can see a detailed list of the steps I followed to derive the reflectance values out of the raw images. There is a dilemma here that one inevitably has to face, and the question is it better to purchase a system that comes with a corresponding processing package, or is it worth building your own software? Again, one should thoroughly consider the scope of his system. So the first solution is expensive, though you have a system that is ready for deployment and that doesn't require you to spend hours of educating yourself on calibration procedures, corrections, and so on. But having a black box makes things a bit harder if you decide to add bits to the processing chain. Obviously, we chose to build our system because we have the labs to perform all needed optical characterization. And yes, it has been a learning curve, but we have been upgrading the system constantly. And now we have a system that I believe exceeds the standards. But don't be disappointed at this point. I think that users can be flexible nowadays because the market is expanding fast. So building a hybrid model maybe is quite feasible today. And there are people like us in RAL Space that can help you build your system or amend your existing one, update your calibration files, and so on. So I know that all this information may sound too complicated, but here are some more details that will help you untangle all this mess, hopefully. So the electric gain and offset correction is a typical, a simple measurement to make. Please note that these measurements should be independent of the general settings of the camera, like focus, aperture, exposure times, and so on. Yes, it is best to get these measurements done after finalizing those settings before the image capture, but it won't have a huge impact if you forget to do so. Here you can see the results for different exposure time setups for a specific band of our Zempila visible imager. So you're done with your first correction. nice and easy. Now, one has to correct for what is widely known as vignetting. And the approach here diversifies based on the platform you are using. So if you're in a lab, you only have to image a homogeneous surface, something not too bright to avoid saturation, and of course not too dark so you can have a significant amount of counts. Usually you can purchase such a reflectance surface by the camera manufacturer, but make sure you order a relatively big one in case you need to move further away from your sample. Here we have a 25 by 15 square centimeters target that worked like charm for our applications. So when you mount your system onto a UAS, then having measurements in front of an integrating sphere is what you actually need for this correction. Here you can see the results we got when we performed this kind of tests in front of our integrating sphere here at RAL Space. And this was an interesting test that helped us also define the optimal aperture size for our measurements. But also it helped to define dead or problematic pixels. And we were lucky, we got none of them. So for the linearity test, one can try several combinations of settings in front of our integrating sphere, but you can also test the linearity of the channels by using our reflectance calibration targets. Either way, the procedure is relatively simple and very beneficial because not only you have an overview of what is happening with your sensor, but you can assess its linearity and make corrections if you need to. Like, for example, if you have a radiance calibration certificate at specific settings and for some reason you have to change the exposure time because your target is getting too bright or too dark, then you may apply your certificate and account for this change through your linearity factors. So my point here is that there is always a way to move forward with this kind of issues. And just because we like to keep our standards high, a check on the spectral responses of the bands is always needed. For this optical characterization, we used a monochromator from the Oxford University, courtesy of Dr. Williams. But hopefully, we will have a new monochromator in our lab in a few months. As you can see here, it is crucial to know the actual response of your bands because this will help you identify where the actual peak of the band is. Something that is quite important when doing your data analysis. And for us, it was a one-way route because we added some high and low pass filters onto the systems from front optics to exclude some spectral features outside our spectral target range. Thus, we had to know that these filters were doing their job. And the final step of this processing chain is the target imaging, a process which again is very significant, and for that reason we have a variety of gray and colorful reflectance reference styles that are used to derive the absolute reflectances and the exposure times as well we should use during the flight. And as we mentioned before, these style measurements are also used to assess both the linearity and the sensitivity of the channels themselves as a test of their stability of the sensors. And finally, after analyzing all these technical items, it's time to see some interesting, colorful images. So we're ready to move on with the application section that we have been working on and that are related to the food sector. I thought that it would be good to split our applications into two major categories, the ones performed in lab under controlled conditions and the ones done on the field, so we can keep up with the different approaches in handling the hyperspectral system and the data analysis. We have done some work on tree and plant diseases in the lab, and in this example, we worked with Scott's pine needles with aim to identify the diseased part of the needle. So what we did was to image the pine needles both with a spectrometer using an integrating sphere, as well with our hyperspectral cameras. So this was actually the first in-lab measurement that Michelle has done in 2018, just before I joined the group. What I did was to develop the calibration procedures and build a needle recognition algorithm in order to analyze the hyperspectral dataset. The results were then processed with the help of Dr. Lund in the Oxford University. And here you can see the confusion matrices from both types of measurements. The spectrometer's data achieve high accuracy on healthy needles, while these measurements were struggling to recognize the diseased ones. On the other hand, the hyperspectral images gave some pretty consistent results on both healthy and diseased samples. So the images got a failure rate of about 20%, which means that the hyperspectral information failed to correctly categorize the diseased pine needles, while this failure rate was raised to 25% based on the spectrometer's data. And that's probably because the hyperspectral images capture the information onto a two-dimension panel, while the spectrometer's data corresponded to a 3D viewing, which probably made things more difficult when analyzing the data. Another test we had the chance to make was to image plants into a glasshouse environment with our fellow colleagues Paul Brown, Lee Butler, and Andy Crow. And this experiment was aiming to see differences in plants that were inoculated with septoria and rust against healthy ones that were used as our control plants. Unfortunately, our control plants got infected as well, so we couldn't conclude with this experiment, but it was a nice opportunity. Thus, I have included this image here to work on the co-registration of the visible and near-infrared images. Since the two images are placed side by side, the two types of images have a spatial shift in the viewing scene. So the measurements were useful after all. We also got the chance to work with some delicious Gala and Braubeern apples that Dr. Rees from Greenwich University has brought to us. The aim here was to see if the hyperspectral images can identify sugars in apples as an indication of their maturity. So what farmers do so far is to randomly pick some apples, cut them in half, and stain them with iodine in order to see if the starch has started breaking down into sugars. Here at the bottom level of the slide, you can see that starch into unripe apples has stained the iodine. Once this process has begun, farmers only have a few days to harvest the apples, which can be a quite stressful and intense period. So what we did was to image both ripe and unripe apple slices, aiming to see differences in the image spectra. We then realized that the water content of the sample was interfering with our results, although we had actually pampered the surface of some fruits because the water droplets were quite obvious. Unfortunately, by that time, the harvesting period had arrived and we couldn't find any unripe apples. So we used bananas as our model system. So we had the chance to eat some bananas too. The only thing we did differently at this test was to dry the bananas before imaging them. There definitely were some challenges as uneven surfaces affected the images and thus our classification algorithms. But the question that arises here is, do the hyperspectral images perform poorly? Well, I wouldn't say that, and here is the reason why. On this slide, you can see spectrometers measurements on the banana samples performed in an integrating sphere, as done previously with the Scott pine needles test. The difference between the ripe and unripe one is not very obvious, as you can see at the bottom two figures. Therefore, I would say that personally, I'm quite pleased by the way that my analysis has worked for this experiment. And now let's move to some field campaigns that we had in 2018-19, just before the pandemic. As mentioned already, our system is designed to perform both in lab and on field. And on the top panel, you can see that the setup of the two Zempila cameras, along with the battery case, the battery itself, and the computer that hosts the software which is handling the image capture. The whole system weighs less than 1.8 kgs and is mounted on a DJI running gimbal onto a DJI Matrice 600 Pro. While this system is also paired with a GPS sensor, so every image has its unique spatial stamp, which is very useful when you're staging your survey images. To monitor changes in the ambient light, we also have a spectrometer measuring on ground, and its readings are used to compensate for changes in the light conditions during the flight. So the question is, what can we achieve by flying a hyperspectral system over woodlands, orchards, crops, and so on? What we have already done with our colleagues in FERA and in conjunction with Rachel Goldon and Elias Berra from Newcastle University, based on the data that we have gathered from our campaigns in the UK, was to create orthomosaics and 3D maps of the areas we flew over. We were also able to test some tree species classification algorithms that were also validated against ground truth. What the plan is for the future is to try to assess the number of trees, the ground size, leaf area, ground coverage, and some key vegetation indices that can potentially be used as inputs into models to calculate, for example, ground primary production, carbon uptake from plants, or other greenhouse gas fluxes, and so on. We are also working on tree health assessment studies aiming to detect mildew and hopefully scab on apple trees. So we had 4 campaigns over an apple orchard in 2021 that were also coupled with ground-based spectrometer measurements and health assessments from experienced agronomists. So we aim to process the data accordingly, and fingers crossed, we may see some mildew on the leaves from our aerial imagery. And in conclusion, there are a lot of applications that make use or can make use of hyperspectral technologies. Some feed the output of the imager directly into the classification algorithm that is tailored for the specific application, while some other applications are using the outputs as proxies of a more complicated system. And with this last comment, I would like to thank you all, and I'm eager to hear your thoughts, comments, and of course, your questions. Thank you.